Have you ever really stopped to think about how a tiny little pill can have such a huge effect on your body? I mean, how does it know where to go and what to do? Well, it's a pretty amazing journey, really. A trip from your hand right down to the microscopic machinery inside your cells. So today, let's pull back the curtain and see how medicines really work.
Let's start with a classic question, one you've probably asked yourself. How does a pain pillar know how to find your headache? Here's the fun part. It doesn't. At all. A drug doesn't have a GPS. It actually travels everywhere through your bloodstream. But, and this is the key, it only springs into action when it finds the very specific cells that are equipped to listen to its message. Let's dig into how that actually happens.
To get a handle on this, you gotta see a drug's life inside you as a two-part story. It's basically a conversation between the medicine and your body, and each one gets a turn to, do its thing. And this slide lays out that conversation perfectly. Over on the left, you've got pharmacokinetics. You can think of this as everything your body does to the drug. It's the delivery service, right? How the drug gets absorbed, where it gets sent, how it's broken down, and eventually how it's kicked out. But the real action and what we're focusing on today is on the right, pharmacodynamics. This is what the drug does to your body. This is where the magic happens. So, what does that moment of action actually look like? Well, the best way to wrap your head around it is with a really simple analogy. Imagine a world of keys, locks, and, even a few clever imposters. Let's see how this all fits together.
So, every cell that a drug is designed to target has something called a receptor. Just picture a very, very specific lock on the outside of that cell. The drug molecule is designed to be the one and only key that fits that specific lock. This is that molecular docking site where it all kicks off. Okay, so when the perfect key finds its perfect lock, we call that key an agonist. This is the master key. It slides in, it turns smoothly, and it fully activates the cell to do its job. A classic example is the albuterol in an asthma inhaler. That's an agonist that finds receptors in your lungs, unlocks them, and tells the airways to relax, making it way easier to breathe.
But what if you want to stop a process from happening? Well, that's when you send in an antagonist. The best way to think of this one is like a key that breaks off inside the lock. It fits perfectly, so it slides right in, but it can't turn. And just by being there, it physically blocks the real key from getting in and doing its thing. For example, beta blockers for high blood pressure do exactly this. They block adrenaline from getting to its receptors on the heart.
And of course, there's a middle ground. Meet the partial agonist. This is kind of like a worn-out, janky key. It fits, and it can turn the lock, but, not all the way. It gets a response, but it's always a weaker, smaller response than what the full master key could do. Some medications for addiction work just like this, giving just enough of a signal to stop cravings, but without producing the full effect.
So here's where it gets even more interesting. Not all these cellular locks are made the same. Our bodies are smart. They use different kinds of receptors for different jobs, and we can sort them into 4 big families. This chart gives us a fantastic look at the whole lineup. First up, you have ligand-gated ion channels. These are like lightning-fast gateways on the cell's surface that just pop open and closed. Then you've got the G-protein-coupled receptors, or GPCRs, which are way more common and kick off a more complex chain reaction inside. Next are enzyme-linked receptors, which, as the name suggests, switch on enzymes to get things done. And finally, you have intracellular receptors, which are actually deep inside the cell and can go straight to the source and change your cell's instructions directly. Each one is a totally different kind of lock, built for a very specific purpose.
All right, so the key's in the lock, you hear that click, now what? The message doesn't just stop at the front door. Oh no, it sets off this incredible relay race deep inside the cell.
This whole process has a name, signal transduction. It's basically the entire chain of events that takes that initial signal, the key turning in the lock, and passes it along, translating it into a real, meaningful action for the cell.
And that cascade, that relay race, looks something like this. The drug binds, which wakes up an initial protein. That protein then creates these things called second messengers. Think of them like tiny couriers sprinting through the cell with a message. These couriers then activate other proteins, which activate others, and so on, until you get the final result you were looking for. So you might be asking, why so many steps? Why not just have the drug do the final thing directly? And the answer to that is this incredibly powerful concept called signal amplification. It is the secret weapon of this whole process, and it explains how one tiny event on the outside can have a massive impact on the inside. Because here's the thing. One single drug molecule binding to one single receptor doesn't just activate one protein inside. No way. It sets off a chain reaction that can activate hundreds, sometimes even thousands of other proteins down the line. That initial whisper of a signal gets amplified at each step, becoming a shout. That's how a teeny tiny dose of a drug can have such a profound effect on your entire body. It's all about amplification.
Now here's something you might have experienced. If you've ever taken a medication for a long time, sometimes it feels like it just stops working as well. Well, the science we've just been talking about explains that too. It's basically a case of the locks just getting worn out from overuse.
This usually happens in one of two ways. On the left, you have desensitization. This is a pretty fast change where the receptor just gets a little tired and less responsive. The lock gets rusty and harder to turn. But on the right, there's downregulation. This is a slower, more deliberate process. The cell basically says, whoa, I'm getting way too many signals, and it starts physically removing the locks from its surface, leaving fewer places for the drug to even bind to.
So, there you have it. From a simple pill all the way down to a complex intracellular relay race. That is the incredible journey of a drug. It's this whole hidden world of molecular keys and locks, all working together to keep our bodies in balance. And it leaves us with a really fascinating question to think about. As our understanding gets better and better, can we one day design the perfect, custom-made key for every single lock in the human body? The future of medicine is writing on that answer.